I would like to thank the organizers and those involved in the generation of this event for this space to be able to contribute a little alongside these international representatives of oncological science in this important task that concerns us all, which is the study and understanding of cancer. My name is Andrea González Báez. I am a professor at the Faculty of Veterinary Medicine and Zootechnics of Universidad Autónoma de Nuevo Leon, Mexico, and I am going to talk to you about a project that we have been working for a while, which is the effect of BRCA silencing on the nervous system in a murine model of breast cancer. First of all, I would like to talk a little about breast cancer, which is the most common cancer worldwide, with a fairly homogeneous distribution in all continents, and is one of the main causes of death in women. In addition to being one of the pathologies that generates greater morbidity in different aspects of life of those who suffer it and also those who survive it, affecting economic aspect as one of the most expensive cancers, social aspect generating isolation and incapacities, as well as physical and mental effects due to the pathology itself and the treatments. Although these numbers are already alarming, the projections are not better at all. Statistics provided by the World Health Organization estimate that by the 2050, the number of diagnosed cases will increase by up to 40%, and deaths resulting from this pathology could increase by almost 70%. Breast cancer can be classified in many ways. Classifications according to molecular subtypes are necessary to adequately define the therapeutic scheme to be managed in each case. Depending on the molecular type, we can divide it into tumors positive for hormonal receptors, which are called luminal, and they are divided into A and B, and they're usually the most common diagnosed. The other subtypes of tumor are the one with positive HER2 receptors and negative for hormone receptors. And the third type of tumor are the triple negative, which does not have identifiable hormone receptors or HER2 receptors. And although is the one that is diagnosed the least frequently, it is the most aggressive molecular subtype, the one that has the worst prognosis and the highest relapse rate at 5 years. It is also this molecular subtype that is the most related to genetic mutations. As we all know, cancer is a disease whose etiology is multifactorial, where genetic, environmental, and pathophysiological factors converge to generate the appropriate biological context for the development of the pathology. The study of this factor has been carried out to meet various objectives in cancer. Particularly in recent years, various genes have been studied that, when mutated, could be involved in the metabolism of cancer. In the special case of breast cancer, various genes have been identified that not only provide a predispositions to suffering from breast cancer, but their detection could imply modifications to the treatment or even provide other resources for the approach, for the therapeutic approach. Particularly in breast cancer, the BRCA1 gene has been one of the most studied. The BRCA gene is a gene found on chromosome 17, and that, under normal conditions, plays an extremely important role in cell regulation and stability, participating in the activation of different molecules that act in different processes, such as the regulation of cell cycle, the regulation of transcription, the ubiquitination cycle, the regulation of chromatin structure. In apoptosis, it functions also as a tumor suppressor gene, and above it all, participates in DNA repair mainly through homology-directed repair. So, when this gene does not function correctly due to a mutation, these processes are compromised, affecting the stability of the genome, resulting maybe in a cancer. Mutations in these genes, as we said in the previous slide, have a direct impact on genomic stability, which causes the cell to replicate uncontrollably, and this increases the risk of developing cancer. More than 12 mutations has been identified in the BRCA1 gene, including insertions mutations, deletions, and missense mutations that result in a non-functional protein, which increases the risk of those who have this mutation of suffering breast, ovarian, and pancreatic cancer, among others. Significantly increasing the risk, with the average in ovaries, for example, being of 2%, and in the case of patients mutated in BRCA1, up to 70%. Up to 87% in the case of breast cancer, compared to the average of the population of 16%. This is why these mutations are called pathogenic mutations, and prophylactic procedures can be performed around this increased risk that can range from the intensification of monitoring protocols, the administration of different kinds of drugs, and even a surgical approach. Cancer resulting from these pathogenic mutations represent 5 to 10% of all cancers, and in the case of breast cancer, up to 3%. We already know the relevance that BRCA1 has in cellular and genomic stability, which does not make it strange that it is also associated with other processes in the body. It can be seen that BRCA1 plays a predominant role in the development of correct functioning of the nervous system, to such a degree that BRCA1 knockout models do not survive, since BRCA1 is especially important in the early stages of brain development. On the other hand, in adult stages, the mutation has been related to different types of neurodegenerative diseases such as Alzheimer, amyotrophic lateral sclerosis, and Parkinson, inferring that the damage caused by age and various injuries could represent a greater risk in individuals with BRCA1 mutations, since the proportion of repair in relation to damage could be affected. Continuing with the study of these factors that interact for the development and metabolism of cancer, it has been identified how the nervous system plays a predominant role in the development of cancer, where there is a bidirectional communications between the nervous system and the tumor. This is not only in tumors within the skull, but even in distal tumors of the central nervous system. It has been seen that the interaction of cancer and the nervous system occurs through different mechanisms, such as paracrine interactions, direct synapses between neurons and cancer cells, emulation of tripartite synapse, the formation of multicellular networks exchanges of ions, perineural invasions, regulation of the tumor microenvironment through innervation, the production of neurotransmitters that are communicated with a tumor, among other mechanisms, which have been shown to actively participate in promoting tumor metabolism. Profundizing with these interactions between nervous systems and cancer, the effects of different cognitive dimensions in cancer patients are well known, compromising memory, attention, problem-solving ability, executive functions, fine motor skills, verbal fluency, among other cognitive skills, have been reported. Traditionally, these effects were attributed to the neurotoxic effect of pharmacological treatments and the side effects of hormonal treatments. However, there are studies that tell us about alteration in these cognitive dimensions prior to the start of the therapeutic regimen, which tell us that the cancer itself can infer and affect the patient's nervous system. And this is based on these known bidirectional interactions between cancer and the nervous system. This is why our work focuses on studying the interactions between cancer patients that has BRCA1 mutations and the nervous system, since we believe that these mutations itself provide a vulnerability to individuals whose nervous system is already affected by cancer alone, which is why we believe it is important to describe this vulnerability so that palliative treatments consider the protection of the nervous system to limit important cognitive sequelae in the patients. The development of our experiment was structured in two phases. The first one, in vitro phase, it's in which we use the 41 cell line which is a spontaneous mammary carcinoma line from BRCA mice, and whose characteristic is to generate triple negative tumors, which are the ones most closely related to hereditary cancer. At this stage, the basal expression of BRCA1 was evaluated, and silencing was carried out using an RNA interference. The second stage is the in vivo stage or phase, where a murine model of breast cancer was established using this 41 cell line in female BRCA mice. In the same way, the BRCA gene was silenced by administering interfering RNA. All groups underwent different ethological tests to evaluate the different cognitive dimensions, and after the endpoint of the animals, histopathological lesions were evaluated, as well as different biomarkers of neuroinflammation, neurodegeneration, and neurochemical functionality. Continuing with the work scheme, I would like to present the progress we have made. We profiled the expression level of the gene that we want to silence, BRCA1 gene, specifically in the 41 cell line, obtaining a significant expression of the BRCA1 gene in relation to its normalizer. This gave us the guideline to start working with this line. Later, we began to standardize the breast cancer model. This has some difficulties because direct inoculation into the mammary gland in mice is often an act of faith, since the breasts in mice are very difficult to see. It is important to refine the management and technique to avoid accessing the peritoneum and leaving it very superficial. Training to guarantee the success of inoculation and implantation of the tumor in the mammary gland is essential. However, by optimizing the inoculation, we were able to confirm successful implantation and observing exponential growth until day 15. We confirmed the implantation of the tumor through histopathological techniques. We were able to observe an increase in the size of the mammary gland due to cell proliferation, as you can see in the first photo on the left and up side of the slide, as well as the presence of round and polyhedral tumor cells in the mammary gland surrounding a blood vessel, as you can see in the arrows and in the right side of the slide, in the upper and right side of the slide. Then later we extract the entire brain from the mice subjected to the cancer model and evaluated on days 7 and 14. On day 7, as you can see in the first photo in the lower row of the slide, we observe no relevant changes in the brain tissue. However, on day 14, which also coincides with the highest peak of the tumor diameter, we were able to observe relevant changes. Here in the second photo, we can observe the cerebral cortex, which has areas of demyelination. By observing these back walls, which are changes related to neurodegeneration. On the other hand, on the same day 14, on the third photo, we can also see changes associated with neuroinflammation, where we can see these glia cells and lymphocytes surrounding this blood vessel. And you can see the arrows pointing it. In the same way, in the last photo, I think that I'm blocking a little bit the view, but we can see satellitosis. We see these glial cells surrounding the generated neurons and beginning neuronophagia. These changes were not identified in our control mice. So we think that the changes may be due to our model of cancer. To evaluate cognitive alterations in the murine model, we used different behavioral tests. One of them was open field, where the mouse is exposed to a box and different parameters are evaluated by tracing its route. Things such as acceleration, distance travels, exploration rate, mobility rate, and the time spent in the centers of the place compared to the periphery are evaluated. We saw some interesting changes in the open field test. The average speed in the treatment group was reduced compared to the control group, especially at day 14, Like also we said, it's also coincide with the size of the largest tumor. The mice in the treatment group at day 14 had a significant decrease in the speed of their movements. On the other hand, when evaluating acceleration, we were also able to identify significant decrease in the mice on day 14 compared to the control mice. The mice look more lethargic and their movements became more deliberate and slow. You can see a tendency of slowing the acceleration on day 14 compared to the control. The total distance traveled was also compromised in the animals that were under treatment compared to the animals in the control group, again to a greater extent on day 14, which we remember coincides with the size of the largest tumor. The animals had fewer movements in general. This lethargy had a direct impact on their distance travel. To evaluate learning and memory, we use the known object and novel object test, where we expose the animal to an object for a particular time to subsequently expose it to a completely new object along with the known one. We count the time spent on each object and make comparisons. In this case, although we did not obtain a significant difference, we can observe a reverse trend in the treatment group in relation to the control group. We can observe that the mice in the control group tends to spend more time with a familiar object compared to the novel object. This is not the case in the treatment groups, where the exploration time with the novel object increases. This may lead us to think that memory could be somewhat affected and that anxiolytic exploration behaviors may also be affected. Finally, I will show you the grip test. This test is intended to evaluate the motor skills of the subject. The animals are exposed to a mesh, trying to get them to grip, and then invert the mesh and count the time it lasts held on its four legs. In this case, although we did not obtain a significant difference, which tells us that the mouse does not have any of its motor functions altered, that make us think that alteration in its behavior are not necessarily due to something mechanical like pain or something that may affect her way of walk. So this result is actually very good for us. I would like to thank you the opportunity